Hi, welcome to Biochemistry 1, also known as CHEM4500. Before we begin, I wanted to take an opportunity here to just sort of give you a brief overview of what it is that we are going to be doing. So, for most of these lectures, you're going to find that I'm going to stick reasonably closely to your textbook. So, you'll find that for most instances, you can follow along with the lecture material by reading in your book. In some cases, I may not cover certain concepts that are maybe not as important for this class. Sometimes your textbook will throw in more advanced concepts that are great, but we don't always have time for that. So for the most part, I will stick to the flow of each of the chapters in your textbook. Chapter 1, however, is an outlier. I'm not going to stick very closely, as you'll see here in a moment. Chapter 1 is really meant as an orientation to biochemistry. And the way that we do that is really by reviewing material that you have seen before. What you're going to find here is that biochemistry is a very interdisciplinary science. But for you, what this means is that you're going to have to draw upon material from various different courses that you have already taken in a way that you probably have not before. So for example, there will be times when in a single lecture, we may have to invoke organic chemistry concepts alongside general chemistry concepts, alongside biology, alongside analytical chemistry. So the point is that what you're going to be doing here is really pulling together information, knowledge that you already have, but in a way that you probably have not been challenged to do so before. So for that reason, this first chapter, I do not really tend to use, I don't stick to the textbook very closely. I rather use it to give you a prompt, an idea of concepts that you may need to review that you've seen before. Because you have seen a lot of these concepts before, but just from a different perspective. So here, I'm going to give you sort of an orientation and at the same time, hopefully prompt you to pick up your old textbooks or go to Wikipedia. Just try to refresh yourself on certain concepts. And I'll try to highlight these as we go. And now, I hope that you have your cup of coffee in hand because it's time to actually start talking biochemistry. Let's go. Before we can go any further, before we can even start, we have to stop and ask ourselves, what is biochemistry? This is a question that you really should already be pondering. Well, the answer is not simple. There's a general answer, which is an answer that I think we can all agree upon. Biochemistry is the study of the chemistry of life, hence the name, biochemistry, chemistry of biological systems. So we are going to be using chemistry to understand biological questions. Now, the approach to do so is rather diverse. And what I mean by that is biochemistry as a field is an incredibly broad field that encompasses individuals with very diverse sets of expertise. If you take a biochemistry course at, say, 3 different universities, your experience may be wildly different. I know of cases where faculty members that are teaching biochemistry are trained as, say, toxicologists, and they teach it from more of a biologist perspective. In fact, biochemistry sometimes shows up in departments of biology. For myself, just to give you a little bit of transparency, I am trained in chemistry, and not just chemistry, but I am trained in biophysical chemistry. So when I address a biochemical question, it is usually from the perspective of physical chemistry. You'll probably see that and get annoyed with that going forward, but I want you just to understand that what biochemistry is highly, highly dependent upon who you're asking. So when I address the fundamental questions, which these are questions that all biochemists can appreciate, for example, how do biological molecules interact with each other? How does the cell manage its energy needs? How are biological molecules structurally organized and related? And lastly here, and this is not meant to be exhaustive, but how is genetic information stored, transmitted, and expressed? So these are four very basic questions. All biochemists, biologists as well, are going to be able to agree that these are important questions. But the approaches, the techniques that are used to answer these questions can very much depend upon who it is that is trying to answer the question. So going forward, I just want you to understand that the way that I teach biochemistry, it is meant to address these big questions, but I am teaching it from my own perspective. So, many of you will be coming from the biology department. A lot of students that take biochemistry here are biology majors. So what I want you to sort of think about is, the how is the perspective I am teaching going to be translated into your field of study? How can we bridge these different perspectives? So before I move on, I just really wanted to introduce to you first what biochemistry is. It is the study of the chemistry of life. Chemistry is at the core of biochemistry, so we are using chemistry, chemical methods, chemical concepts to address biological questions. And I want you to understand that the way that we go about doing this is highly dependent upon who it is that is undertaking the study. So as we move through this course, one of the things that I want you to remember is context. So as we talk about various concepts that may seem very chemistry-y, what I want you to remember is that we are always going to be thinking about these biochemical concepts in context. And the context here is the cell. So when we talk about biochemical observations, usually we are making them in an extracellular in vitro context, and we are going to try to translate, or rather extrapolate, those observations to a cellular context. Now, just to give you an example, or a point of reference here as to why context is important, this is a dated example, but one that I hope still works just a little bit. Imagine you are walking down the street and you observe a guy with a hockey mask on and what appears to be an axe in his head. For those that don't know, this is Jason Voorhees, a picture of him from the Friday the 13th movies. And again, that's a dated reference, but if you don't know, go look it up. So this would represent my experimental observation. And if the frame I'm showing you is all that you saw, you might freak out a little bit. You might say, I should probably be concerned because either there's a guy with an axe in his head and he needs help, or, the other, takeaway might be, there's an undead serial killer that's going to try to get me. Either of those require some action on your part. Now, this might change entirely if you had a fuller picture and you realize that you somehow stumbled upon, maybe it's Halloween, maybe it's you're in Hollywood on a movie set. How you got there from Tennessee without knowing it, I don't know. But you stumbled upon something that's a lot less threatening. You have a guy dressed up as Jason Voorhees sitting in a chair reading what appears to be Time magazine. So this is a lot less threatening, but this is your context. So as we move forward, obviously, I hope obviously, not everything that we're going to talk about is going to come back to horror movies, but the big takeaway here is that I want you to try just to view these concepts that we're going to talk about in a biological context. Remember, we're going to be talking about chemical concepts, chemistry applied to living systems. So sometimes connecting the dots may require you to really just sit down and try to think about context. So just keep this in mind. If the cell is our context, let's take a moment and just appreciate what a cell is. Well, a cell is defined in different ways, but fundamentally, it is an autonomous, self-replicating unit that may exist as a functional, independent unit of life. So as you can see here in this GIF, you start with a few cells, then propagate. They divide into many more cells than were there initially. They are autonomous, self-replicating units. They have within them all of the machinery that allows for replication. The second part of this is that they are membrane-bound structures that contain biomolecules that represent that machinery. These biomolecules are nucleic acids, proteins, polysaccharides. So these biomolecules are really where we are going to, in this class, spend all of our time. What we are doing in Biochemistry 1 is laying the foundation. We are going to spend most of our time talking about the biomolecules themselves. This is the foundation. What do they look like? What do they do? How do they behave? And so before I move on, one thing here that I want to point out, and this is a question I get every time I teach this class: are viruses living organisms? And my answer to that is no, they are not. And that is because they do not satisfy the first point here. They are not self-replicating units. They require a host to replicate. So viruses do not fall into this category. They are not autonomous self-replicating units. So there is some, something to discussing proteins that come from viruses in a viral context. But again, that requires a different perspective. Most of what we are going to be talking about is related to proteins, polysaccharides, nucleic acids that come directly from the cellular context. So just please try to remember this as we go. Cells are highly compartmentalized. They have membranes that allow them to achieve very specific goals. So first, of course, you have your outer membrane, and this basically keeps all of your stuff inside of the cell. This is important because if you didn't have that, your stuff could diffuse out and you really have no order. Think about this, think about a fence that keeps your dog in the yard. If you didn't have the fence, the dog could roam off. So here, the outer membrane keeps all of your stuff inside of the cell so that they don't diffuse out, and really, what do you have then? You also have compartments within cells that are defined by membranes too. Here I'm showing you 4 different compartments, A, B, C, and D. These are important because what this does is it ensures, in a lot of cases, that certain metabolites are isolated to certain parts of the cell and really keeps the concentrations of those metabolites locally high. If you think about the volume of the cell is much higher than the volume of each individual compartment. So if I have the same number of molecules of A in this compartment, the concentration is going to be higher compared to the situation where the same molecules of A spread out over the entire larger volume of the cell. In another view, you can sort of view this like a house. You have different rooms in a house. Your kitchen has your kitchen stuff, your pots, your pans. You want those all to be there in the kitchen for when you need them, as opposed to spread all over the house. So you can more efficiently function, you can more efficiently cook your breakfast if all of your parts, your pots and pans, are in that room. So here, towards that, compartments also isolate specific metabolic activities. So instead of having all of the parts spread out over the cell, and then when you need to do whatever function that is, you have to pull them all to, all of your parts to one area of the cell. Instead, all of your your enzymes, your metabolites that are important to perform some sort of activity are already where they all need to be. So compartmentalization is really important for a number of different reasons, but in particular, maintaining locally high concentrations of metabolites as well as isolation of specific metabolic activities. Going back to high school biology, there are two types of cells. There are eukaryotic cells, there are prokaryotic cells. The main difference is, does the cell have a membrane enclosed nucleus or not? So eukaryotic cells have a membrane enclosed nucleus. There's a nuclear envelope that restricts flow of materials in and out of the nucleus. Our genetic material is sequestered inside of the nucleus, and eukaryotic cells tend to be larger and more complex than prokaryotic cells, which lack a nucleus and lack the same membrane-defined structure. That doesn't mean that there's not organization in prokaryotic cells, but certain membranes are lacking. And so you can clearly see in eukaryotic cells that there is a membrane-enveloped, membrane-enclosed nucleus. These are HeLa cancer cells, and these are from a eukaryotic source. Not really going to go into that history right now, but you can see just from a microscopy image here that's been stained in a certain way that there is a clearly identifiable nucleus. Okay, so I just told you that prokaryotic cells lack a defined membrane-enclosed nucleus. So they lack defined compartments, but for reasons that we're not going to really talk about here, they still are able to facilitate metabolic function in very defined cytoplasmic regions of the cell. In fact, if you look at just a schematic of E. coli, Escherichia coli, you'll find that the nucleus, there's not a nucleus, but your genetic material still sort of sequesters to form what is called a nucleoid. So there is still some compartmentalization in prokaryotic cells, but it just doesn't look exactly like it does in a eukaryotic cell. Eukaryotic cells are much larger than prokaryotic cells. If you don't believe me, let's look at the numbers. So in general, a eukaryotic cell is going to be anywhere from 10 to 100 micrometers or microns, that's 10 to the minus 6th meters in diameter. So that's pretty large. If we look at the volume, eukaryotic cells are 10 to the 3rd to 10 to the 6th times the volume of the typical prokaryotic cell. And this should really make sense to you, because instead of packing all of your stuff into one volume without any sort of compartmentalization, now the eukaryotic cell also contains your membrane-enclosed organelles, which occupy more volume. So there's more stuff that we're trying to cram into a eukaryotic cell, more different machineries for metabolic regulation, and really the complexity in a eukaryotic cell goes up. But if you want to view this simplistically, in the eukaryotic cell, we have membrane-enclosed organelles, which are shown here. Like here you have your endoplasmic reticulum, mitochondria, your nucleus. These are all things that are membrane enclosed. So those additional membranes, the compartmentalization does require more volume. So this is a very important difference to take note of that differentiates eukaryotic from prokaryotic cells. Viewing this a different way, we can look at the complexity of eukaryotes versus prokaryotes from a genome-based perspective. So you'll find that most single-celled organisms have far fewer genes than multicellular organisms. I'll point out here, you've got Mycobacterium, an archaeal bacterium, and E. coli shown right here, which are all single-celled organisms, and they have approximately 500 to almost 5,000 genes for each of these. Now, if you look at the next example, Saccharomyces cerevisiae, baker's yeast, it's also a single-celled organism, but it is a eukaryote, hence the reason it is used as a model organism for human questions. But yeast here still has about 6,000 genes. So we are sort of working our way up in terms of complexity. We go from about 500 genes to 6,000 genes. But these are all single-celled organisms. Now, as soon as we start moving into multicellular organisms, which are all going to be eukaryotic in nature, the complexity goes up quite a bit. So here, C. elegans, these are worms. You have the average organism having just shy of 1,000 cells, but per cell, you got about 20,000 genes. If you go down to humans, Homo sapiens, you'll find we have quite a bit more in the way of cells, number of cells per organism, but per cell, we still have about 21,000 genes. Now, you'll see here that eukaryotes tend to be more complex genetically. We have more genes which encode for more proteins. 21,000 genes presumably gives you thousands of proteins it could be encoded for. That does not mean that they are all present at the same time, but there's a lot more complexity in eukaryotes relative to prokaryotes. Now, I do want to note, though, that more genetic material does not necessarily equal more complexity. So I'm showing you here a picture of a rare Japanese flower that is named Paris japonica. So it has a genome that is comprised of 149 billion base pairs. It is 50 times the size of the human genome. Its genome is 50 times the size of the human genome. It's the largest genome ever found. Second to that, the biggest genome belonged to a marbled lungfish that has 130 billion base pairs. So we're looking at this, and I hope that you see what I see. I see a flower. It doesn't appear to be as complex as humans. So I'm showing this because I want you to realize that more genetic material does not automatically make you more complex of an organism. Humans, in my mind, are more complex than the stationary flower. Okay, so here we've set the context, and this really concludes as far as I'm going to go into the biology. Remember, I'm trained as a chemist, not a biologist, so I'm trying to set the context for you. But now we're going to turn our attention to the chemistry. So let's go straight to the thing that is most associated with chemistry. Let's talk about the periodic table for a moment. When we think of biological molecules, what are they made of? So despite what you learned in general chemistry, we are not going to be concerned with all 118 elements on the periodic table. In fact, the majority of them are not really observed biologically. Instead, we're going to be concerned with a very small number of elements that include carbon, hydrogen, oxygen, nitrogen, phosphorus, calcium, sulfur. So these together account for about 97% of the dry weight of the human body. Now, remember, dry. So if I remove all of the water, this is really what the composition looks like. So you're about 62% carbon, 11% nitrogen, some oxygen, and you can sort of see that you drop off and sort of enter into the trace metals. So you'll see there are transition metals here that are highlighted. These are trace metals. Selenium shows up every now and then, iodine, but the ones that are shown in red, or salmon, if you want to be technical here, these are what we call the bulk elements. So you have a lot more of them, a lot more. They represent more of the dry mass relative to the trace elements. Now, just to put this into perspective, under normal conditions, you and I as humans are about 70% water. So when we talk about the dry weight, it is very important to remember that is the dry weight. Otherwise, most of what you and I are made of is water. Okay, I get it. We are carbon-based organisms. But where's that carbon distributed? Okay, so let's dig a little bit deeper. If we look at this table, you'll see, again, if we look at the total weight of the cell, percentage-wise, you'll find 70% of the cell is going to be water. Yeah, okay, I just told you that. But the remaining materials can be divided between proteins, nucleic acids, polysaccharides, lipids, and inorganic ions. So the majority of that percentage weight is going to be protein. So about 15% of the weight of the cell is derived from proteins. And you'll find that there are a lot of different kinds of proteins. And for reasons we'll talk about later in this course, proteins can be very diverse. And you'll see why that is when we move into later chapters. But this contrasts water, where there is only one kind of water. Water is water, it's H2O. But in E. coli, which is what I'm showing you here, you have about 3,000 different kinds of proteins. Okay, now if we move on down, you'll find 4 nucleic acids, deoxynucleic acids, so DNA, or ribonucleic acids. Actually, that's deoxyribonucleic acid. I just got my language mixed up. But DNA versus RNA, 1% and 6%. You'll see there's a lot more diversity when it comes to RNA. Again, this will make sense as we move into the course. Polysaccharides are another 3%. They show some diversity, 10 different kinds on average per cell. 2% are lipids. There are different kinds of lipids as well. And we'll just skip over this part, but ions, inorganic ions. These are ions like calcium, magnesium. They're about 1% of the weight of the cell, but you've got about 20 different kinds. These are 20 different inorganic ions that you'll find commonly in cells. Now, before I move on here, I've just basically broken down how we're going to spend our time in this course. In Biochemistry 1, we devote all of our time to really presenting to you the different kinds of biological molecules. So we're going to talk about proteins, we're going to talk about nucleic acids, polysaccharides, and lipids. So basically, these are the 4 most common types of biological molecule, and you should see, based on percentage weight in the cell, why we go about doing this. But that's sort of how this course is going to be broken down. We're going to spend time talking about each one of these common biological molecules. The commonality of all of these biological molecules is that they are carbon compounds. They are centered, designed around carbon and its chemical behavior. So carbon is quite versatile in that it can form stable covalent bonds through electron sharing. So this occurs by orbital overlap. Again, chemistry, I'm a chemist. So we are going to view this from a chemistry-centric perspective. Carbon can form as many as 4 covalent bonds by sharing the 4 electrons in its outer shell. So if you look at this, this can occur in many different ways. So this can, you can form the carbon-hydrogen single bond. So this would just be, 1 electron coming from carbon, 1 from hydrogen, and now you have a sigma covalent bond. This can also occur with oxygen. You can get yourself a sigma bond, a just single orbital overlap. You can also form double bonds. So in the case of carbon and oxygen, you can also have 1 electron being shared in a common sigma bond, but you can also have a secondary orbital overlap that occurs through pi orbital overlap to give you the double bonds. Carbon can also form triple bonds, and carbon can form covalent bonds with a diverse number of different kinds of elements. So hydrogen, oxygen, nitrogen. Phosphorus is also a possibility. Sulfur. So carbon is quite versatile in that it can form very stable covalent bonds with a lot of different kinds of other elements. So it's pretty versatile in what it can do. If you don't believe me when I tell you that carbon is versatile, let's look at the commonly observed functional groups that show up in biological systems. So here I'm showing you a summary of all of the common functional groups that you will encounter in this course. So you'll find methyl groups, ethyl groups. You'll find your carbonyls, like aldehydes or ketones. But you'll also find primary amines, or in some cases, secondary amines. You'll find amides. You'll find something called the guanidinium group. You'll find imidazole. So you're going to find that the functional groups that we encounter in this course are diverse. Now, I'm not showing you this to prompt you to go memorize all of them. You'll pick them up as we go. When we get to the discussion of the amino acids, it will be important for you to be familiar with many of these functional groups, but you don't have to do this now. But one thing I do want to impress upon you at this point is that the diversity in functional groups allows for enzymes to engage in very diverse types of chemistries. Certain functional groups are better for the performance of certain chemical reactions. When we talk about protein three-dimensional structure, some of these functional groups will interact with other functional groups in such a way as to stabilize that overall structure. As an example, you will find often primary amines, which are going to be protonated and cationic at physiologic pH, interacting with carboxylates, which are going to be anionic, deprotonated at physiologic pH. So this is an electrostatic interaction. Positive charge interacts with negative charge. And this electrostatic interaction often is observed in stabilization of protein three-dimensional structure. So I really want to impress upon you the importance of recognizing the types of chemistries that these different functional groups can engage in. I'm not trying to overwhelm you at this point. I don't want you to think, oh, crap, I have to memorize everything from organic chemistry, all of these functional groups I've long forgotten. No. You'll pick them up as we go, but just be prepared to do so. Biomolecules universally contain different types of chemical bonds. Many of them are going to contain different functional groups as well. And the point in doing so, the point in combining different functional groups into a larger molecule, really is to increase the chemical versatility of that molecule. As an example, let's look at acetyl coenzyme A. Here, I'm showing you the space filling view, but chemically, you'll see here that there are a number of different functional groups present. There's a thioester, there's an amido, there's another amido, there's a hydroxyl group, phosphoanhydride, there's an imidazole-like group, there's an amino group, there's a phosphoryl group. But you can actually break this molecule down in a little bit more simple of a way. This portion that I am circling with my cursor is actually very similar to a nucleotide. It looks like ATP or something similar. Again, trying not to go into too much depth. So we can break down this complex molecule into smaller portions which still have multiple functional groups present. But the point here is to really get you to start thinking about the modularity of biological molecules. If you have a functional group that does one that has, it is capable of a certain type of chemistry, and then you tack it into a larger molecule that has other functional groups, you basically, if you view this like a Lego sort of approach, you're putting together these modules that collectively give you some desired behavior. Biological molecules are often also polymers. So you'll see that you have individual building blocks that are going to undergo condensation reactions to give you a larger molecule. So here I'm showing you two different functional groups. There's the carboxylic acid shown here and the primary amine here. When we get to the discussion of amino acids, you'll see that basically what I have here is one amino acid and a separate amino acid. These can undergo a condensation reaction to link them together. So the former individual amino acids now are covalently tethered together via an amide bond. This is just one example, but biomolecules tend to be polymers, and the theme here is that tethering together individual building blocks gives you a polymer that behaves distinctly different from the collection of individual free building blocks. One exception here that you'll see going forward are lipids. So lipids don't undergo these condensation reactions in quite the same way. In fact, they don't undergo condensation reactions to form polymers like we're talking about here, but they can still assemble into larger structures that have different properties. So 3 out of the 4 biological molecules that we'll talk about here, proteins, nucleic acids, polysaccharides, these are all polymeric in nature. One thing to note here is that this reaction is shown as a reversible reaction, but it is not freely reversible in living systems. So proteins don't just undergo spontaneous hydrolysis to undo what has been done. They don't just spontaneously fall apart. That would be a major problem because proteins live in aqueous solutions. So we don't actually want that to be freely reversible in living systems. Biomolecules also display structural hierarchy when we talk about them in the context of the cell. Again, you're going to see that we're going to spend a lot of our time talking about the basic biological molecules. We're going to look at nucleotides, which are the building block for DNA, amino acids, which are the building blocks for proteins, monosaccharides or sugars, which are the building blocks for polysaccharides. So we are going to first talk about the monomeric units. These are the building blocks, the subunits. So every time we talk about one of these types of biological molecules, the discussion is basically going to go like this. We're going to talk about the building blocks first. And again, these are your common building blocks. And then we are going to transition to discussion of the polymer. What happens, what does the molecule look like once you have tethered together building blocks into a larger polymer? So nucleotides build into DNA, or RNA for that matter. Amino acids are used to make proteins. Polysaccharides are a little bit more diverse, but we're going to talk about monomers and then the macromolecule, the polymer that results. Now, we will not talk as much about what happens after that. So you'll see in the cell, your DNA is going to undergo packaging to form chromatin, which can then be packaged into the nucleus. So there are different levels, hierarchical levels of structure that occur in the cell. Proteins can assemble into larger complexes as well. Polysaccharides in the cell are used to make, say, the cell wall. They're used to make other things too, but I want you to understand that there is a structural hierarchy here. And in this course, really our focus is going to be on the first 2 levels that are shown here, the monomer and then the basic polymer. The structural hierarchy that we just talked about is really dependent upon non-covalent interactions. As we're going to see, biomolecular structure is going to be driven by non-covalent interactions, and these are really interactions that are occurring between functional groups within each of the individual molecules. These non-covalent interactions are all going to be, with very little exception, weak chemical forces, and they are individually going to be weak non-covalent interactions. Think van der Waals interactions, the hydrophobic effect, electrostatic interactions, hydrogen bonding, dipole-dipole interactions. So these are weak individual interactions, but in the context of a larger macromolecule, there are going to be many of these individual interactions that collectively contribute to the stabilization of overall structure. So I'm showing you a structure here. This is a protein structure. Each individual protein molecule is colored differently. So there are 6 different protein molecules. Each individual molecule has a structure that is stabilized by a network of weak non-covalent interactions. But then each protein interacts with other proteins to stabilize an even larger, more complex structure. So biomolecular interactions, these weak non-covalent interactions, drive structural properties. That is, they drive the 3-dimensional structure that is formed. An incredibly important part of these non-covalent interactions is that they are reversible. They are not covalent interactions. They are non-covalent, therefore they are readily reversible. Now this is really important because biomolecular interactions, they need to be transient in nature. They need to be forming and breaking and forming and breaking. And the reason for this is it allows for protein flexibility. Enzymes, I'm showing you an example here. This is a helicase that is moving along DNA. They must be flexible. They cannot be flexible if these biomolecular interactions, these non-covalent interactions, are permanently formed. So they basically have to be turned on and off to allow for protein flexibility, or just more broadly, enzyme flexibility. Rigid, static structures would not be compatible with enzymatic function. As you'll see later, enzymes move around a lot. And so we need to use non-covalent interactions that allow for this movement. Okay, so at this point, this brings up another set of material that you may want to review. You may want to look back over your discussions that you've had in general chemistry of non-covalent interactions. These are going to be very important later in this course, especially when we start talking about protein three-dimensional structure. You'll find we're going to invoke non-covalent interactions pretty regularly. So it would be really helpful for you to at least be semi-fresh on what van der Waals interactions are, or what ionic interactions are, things like that. Now we're going to turn our attention to some thermodynamics. These are concepts that you have learned in general chemistry, but I'm going to just briefly review them just to bring you up to speed. Okay, first, let's throw out a definition. What is thermodynamics? What is the field of thermodynamics? Well, it is the branch of physical science that deals with the relations between heat and other forms of energy. In particular, we're going to focus a lot on chemical energy. So what we are going to do is use thermodynamics in a chemical context most of the time. Now, this definition that I just gave you, it's kind of vague. It is very all-encompassing. So let's be a little bit more specific. We are going to be interested in the use of thermodynamics to address the following issues. The first of which is the ability to predict whether or not a chemical reaction is going to occur under a specified set of conditions. This is incredibly important when we start thinking about enzymatic function and metabolism. So chemical reactions in cells are individually described by thermodynamic properties. So we can use those properties to predict whether or not a reaction is going to occur. Now, one thing to state here is that we cannot predict how fast a reaction occurs using thermodynamics. Thermodynamics only allows you to predict whether a reaction will eventually occur. It says nothing about the kinetics of the reaction. Now, the second thing here is if a chemical reaction or phase change occurs, what are the energy changes associated with that process? So how much work can be produced? In other words, how much energy do I have to either put in to make a reaction occur, or how much energy would be produced by a reaction? This particular point is very useful when we start talking about metabolism, because you will find reactions that are coupled to one another. And this all boils down to the thermodynamics. Lastly, if a chemical reaction occurs, to what extent does the reaction occur? So what is the position of the chemical equilibrium? What I mean by that is, remember, equilibria is simply we're only concerned with how much product is formed relative to reactant. So what is the ratio of product molecules or product concentration to reactant concentrations? So really, if a chemical reaction occurs, how much more product would I produce? What is the position of the chemical equilibrium? What is the ratio of starting material and product? Okay, so hopefully as we move forward in the class, you'll sort of see how these issues play out. But up front, I want to go ahead and just tell you that when we talk about thermodynamics and biochemistry, at least in this course, we're not going to be diving into, quantum mechanics or, real deep physical chemistry. We are trying to use thermodynamics as a tool to understand chemical processes that are occurring in the cell. So thermodynamics here should not be viewed as something that is unnecessarily quantitative. It's something that we're going to use as a tool. My goal is to get you to be able to interpret thermodynamic parameters and make statements and predictions based off of those parameters, not necessarily do math for the sake of math. So please just understand that going forward. All of the thermodynamic parameters that we're going to talk about in this course are considered to be state functions. So a state function is a thermodynamic quantity whose value depends only on the state at the moment. Now, what does that even mean? What does that mean at all? What that means is thermodynamics state functions don't really care about your path. They don't care how you got from point A to point B. So you'll see here, point A, point B. In the first path, it's just straight. A connects directly to B. So this is a straightforward path. But you may also take something that's a bit more circuitous. Maybe what we're describing is my trip from my office to the bathroom. Maybe I go straight to the bathroom, but maybe I walk down to the first floor of the science building, I meander a little bit, come up some stairs, get lost in a classroom somewhere. Maybe I bust up into a lecture class that is ongoing and I just sort of get lost. But ultimately, I find my way back up to the third floor of the science building where the bathroom is, and all is well. So if you were using thermodynamics to describe my path to the bathroom, you wouldn't care how I got there. You would only care that I got there. So when we talk about chemical reactions, all we care about is reactant and product. We don't care about what happens in between. So when we think about this, the question really is going to be, how much product do I have relative to reactant? And it's that ratio that we care about. We really do not care about anything in between because thermodynamics are state functions. They are path independent. Now, to really hammer in another point, kinetics are not thermodynamics. So when we talk about A to B, these two paths, as I described them, going from my office to the bathroom, hopefully we can all appreciate that it is going to be faster just to go straight there. But the thermodynamics do not tell me anything about how fast a reaction occurs. So when we start talking about thermodynamic spontaneity, that does not tell you how fast a reaction occurs. It only tells you how much product that you are going to have relative to reactant. It tells you something about spontaneity, but it doesn't tell you how fast the reaction is going to occur. We are now going to take some time to walk through the laws of thermodynamics. Now again, I want you to remember, I am not trying to make you an expert on thermodynamics. I'm not trying to make you learn all of the mathematical ins and outs of thermodynamics. I'm basically giving you a qualitative picture here just to really get you into the mindset of thinking about thermodynamic parameters, because at the end of the day, I want you to be able to interpret the parameters. I want you to be able to interpret an enthalpy, an entropy, a delta G. I want you to interpret these things, not necessarily derive them in great depth. Okay, the first law of thermodynamics, it states that the total energy of a system is conserved. That is, it cannot be created or destroyed. So that energy has to go somewhere. If there's an energy change, that energy has to go somewhere. Energy is not created or destroyed. It is constant. Now, if we view this in different ways, we have three different systems that we can define. We have an isolated system where we have our system, basically the stuff that we are observing. Imagine, this is a solution of water, and it is in a container where it is surrounded by, in physical chemistry speak, it is surrounded by the universe. It's the rest of the universe. So the system is the part of the universe that you are interested in, and then the surroundings are basically the rest of the universe. So in the isolated system, we have no exchange of matter or energy. So the energy that is in your system is going to be constant. There's no exchange of energy across this barrier. Also, it is closed, so there's no exchange of matter either. So basically, it's like, the coffee container that you can't drink out of, which would be incredibly frustrating. I have a coffee, like a travel mug that has coffee in it. It is insulated, so my coffee isn't going to get cold, but I also can't drink it. There's no exchange of matter. It is permanently closed. So that is sort of the example of the isolated system. In the closed system, however, energy exchange may occur. So your system is closed in that you don't get exchange of matter. Again, I have my coffee mug analogy. There, it's not really well insulated, but it's closed. So, my coffee will get cold. How I got my coffee in there, I don't know, but my coffee will get cold, but no water evaporates. Matter stays within your system. In the open system, and you can think about this like an open coffee mug, there's no lid, so matter can exchange. Your coffee can also get cold, so energy exchange can occur as well. So these are basically the three different types of systems. And if you want to view this in a physiologic context, we are very much an open system. So you breathe. I hope you all breathe. Matter exchanges in that way. You can get cold. So there presumably is the possibility for energy exchange as well. Now, it is a little bit more complex than, I shouldn't oversimplify, but there is an application of the first law of thermodynamics in physiologic contexts. The second law of thermodynamics tells us about entropy, which is from the Greek meaning to give direction to. Entropy allows us to assess whether one state is accessible from another by a spontaneous change. So what do I mean by spontaneous? Well, a spontaneous process occurs without some outside influence. So this means, for a chemical reaction, that I don't have to put energy in to make it happen. Alternatively, you could view this like rolling a ball down a hill. You don't have to put in any sort of energy input to make that happen, and gravity takes care of that for you. Now, the second law of thermodynamics tells us that spontaneous processes are characterized by the conversion of order to disorder. In other words, the universe tends to favor towards disorder. If you don't believe me, grab a handful of marbles, drop them on the floor. They're going to go all over the place. They're going to just tend towards disorder. They are not going to spontaneously rearrange some ordered lattice, some ordered array on the floor. They're going to go everywhere. Okay, another example. Imagine you have a thing of water. You're in your kitchen at home, you have a thing of water. Water here would be the blue spheres. You drop in food coloring. That's being shown here as orange. They are not, in that example, your food coloring is not going to stay as a single layer, and your water is not going to stay ordered like shown here. What's going to happen is, due to Brownian motion, your food color is going to diffuse throughout your water, and everything is going to sort of intermingle. It's going to be somewhat disordered. So that is what we mean when we talk about the universe tending towards disorder. And when we talk about this, what we're really saying is that the universe favors entropy. When I say entropy in this course, I want you to associate that with order versus disorder. A higher entropy equals more disorder. We can relate the first and second laws of thermodynamics by way of the Gibbs free energy equation, named after J. Willard Gibbs, shown up in the right corner here. This equation is delta H minus T delta S, and it'll equal a quantity, a thermodynamic quantity that we'll talk about in a moment. Before I get to that point, I want to sort of break this down for you. So delta H, This is enthalpy. You can think about this as heat. Anytime you see the delta sign, this should tell you that you have a change in something. This is a difference. So if you think about thermodynamics as state functions, remember we are interested in the beginning and the end, the start, finish, point A and point B. So the delta H would be the enthalpy or heat at the beginning relative to at the finish, at the equilibrium state. So delta H here is basically your change in heat. This is where your first law of thermodynamics comes in. This is minus T, your temperature, times delta S. Delta S is the change in entropy. So entropy at the beginning of the process versus the end of the process. Now, if we look at the units here, your delta H is going to have units of joules. Okay, delta H is now, delta H minus T, which is a temperature. This temperature is expressed in units of Kelvin. So here you have Kelvin times delta S, which has units of joules per Kelvin. The kelvins are going to cancel. We wind up with joules minus joules, and this is an energy term. So this leads us to the full Gibbs free energy equation. Delta G is my Gibbs free energy change. Remember, it's not just the Gibbs free energy, it's the Gibbs free energy change equals delta H minus T delta S. Okay, so there's a lot that can be taken away from this equation. This is, as presented, a very straightforward way to view the Gibbs free energy change, but this can be very quantitative if you want it to be. We're going to leave it at the level of interpretation. So if I look at the delta G, if I have a positive delta G, this tells me I have an endergonic process. So endergonic, I have to put in energy to make this process happen. This is non-spontaneous, and I would have to put in some external free energy to make the reaction or process occur. If I have a delta G that's equal to 0, remember the delta represents here the difference in free energy values at the start and the finish of the process. If I have a delta G of 0, that means that my free energy is the same before and after. So that tells me I'm at equilibrium. So there, my forward and reverse reactions are exactly balanced. I am at equilibrium. There's no significant change in the, how much product I have relative to reactant. It's at an equilibrium. If I have a negative delta G, I have an exergonic process. This is a process that is spontaneous. I do not have to put energy in to make the reaction go. It just happens. I start forming product. There we go. So when we talk about delta G's, I want you to try to remember this. When I show you a delta G for a reaction, I want you to immediately be able to look at that and say, that is thermodynamically spontaneous or non-spontaneous or at equilibrium. But the thing that I want you to also know is that if I give you a delta G, and I say I give you a very negative delta G, I ask you, is this reaction going to occur? Okay, the answer would be yes. I then ask you, how fast would that reaction occur? Okay, there you need to stop and say, wait, you gave me a thermodynamic property, not a kinetic property. I cannot tell you how fast a reaction occurs based on the delta G. Please, you'll hear me get up on my soapbox on a regular basis to make sure that you know that there is a difference between the kinetic and thermodynamic interpretations. They together give you a balanced picture of what is going on, but please just remember that your delta G does not give you any kinetic information. To think about this using a mechanical example, let's envision raising a box up an incline versus down. So if I start at the bottom of the incline, I have a box, I have to put in some work to make this box go up. So this would be an endergonic process. My delta G for that process would be positive. Now, once I bring it to the top of the hill or the incline, I let it go and gravity will take care of the rest. The box, the weight will just fall down the hill, assuming that friction doesn't slow it down too much. There, I have an exergonic process. The weight box is moving. I don't have to put in the energy to make it happen myself. So that's an exergonic process with a delta G that would be negative. There is also something to consider here, which is the idea of standard state conventions. If you learn this in Gen Chem, this should be somewhat familiar to you, but when we talk about free energy values, enthalpies, entropies, often what we do is we define them under some standard set of conditions. These are standard state conditions. Physical chemistry convention would state that your reaction would need to occur when your temperature is 25 degrees Celsius, 1 atmosphere of pressure, and your solute has an activity of 1, which often equates to a concentration of 1 molar. Well, these are conditions that are not met so commonly in physiologic conditions or in living systems. For example, most cells grow at 37 degrees Celsius, not 25 degrees Celsius. In fact, some extremophiles grow at temperatures that are near the boiling point of water. So here, these are thermal vents, and there are organisms that grow in here at near the boiling point of water. So clearly, this physical chemistry convention doesn't always work. And as you'll see later, actually, if you take Biochemistry 2, you'll learn about this in more depth. There are modified ways to express the Gibbs free energy equation as well as equilibrium constants that account for biological conditions that deviate from these standard state conventions. The last point I want to make is that your equilibrium constant is related to your Gibbs free energy change. I think that we have all heard the expression at some point in your chemistry career that delta G equals minus RT times the natural log of your equilibrium constant. R there is your gas constant, T is your temperature, but minus RT log K is an expression that gets thrown around a lot. We use it a lot in general chemistry. Now let me actually show you how this comes to be. Where did this expression come from? Well, if we write out a reversible equilibrium as shown, the reactants A and B will combine to give you products C and D. And they do so with the given stoichiometric coefficients. So A molecules of A will react with B molecules of B, and so on and so forth. Okay, so we can write out an equilibrium expression for this, and ultimately you'll see that my equilibrium constant can be defined in this way. So my products, the concentration of my products over the concentration of my reactants, or rather the product of my product concentrations over the product of my reaction concentrations, all of them are raised to their stoichiometric coefficients. And so here ultimately my equilibrium constant is just the ratio of product to reactant. So this is something that often students really overcomplicate. You try to make the equilibrium constant into something a lot more complicated than it needs to be. All it is a ratio of product to reactant. If I have a lot of product, a lot more than I have of reactant, my equilibrium constant will be high. And we're taught in general chemistry that equilibrium constants that are greater than 1 are predicted to be thermodynamically spontaneous. So a reaction that's going to form a lot of product is going to have a high equilibrium constant. Now, if you have an equilibrium constant that is less than 1, that would mean that you have more reactant than product. And based on the fractions here, or the fraction here, this is just a ratio. You should be able to see this. Okay, so now this equation, it factors in to our discussion of Gibbs free energy changes. The actual equation for the Gibbs free energy change, delta G, looks like this. We have delta G with a little degree sign. This is my standard state delta G. So this is my free energy change that occurs at 25 degrees Celsius, at 1 atmosphere of pressure, activities of 1. So that is under my standard state conditions. Now, I already told you that biological systems tend to deviate from those conditions. So this term here, the RT log K, that is a way to correct for deviations from my standard state conditions. Now, the interesting thing here is that at equilibrium, my actual delta G, I've already told you this in previous slides, my actual delta G is going to be equal to 0. That means I am at equilibrium. So under those conditions, this delta G term becomes 0, and I can rearrange the equation to give delta G, this is actually my standard state delta G, but my delta G equals minus RT, there should be a log in here, but minus RT log KEQ. So that's where this expression comes from. Now, okay, so the point of your equilibrium, the point of equilibrium for a reaction in solution is a function of the standard state for energy change for the process. Hopefully, this is clear to you at this point. But one thing that I really want to urge you to do is, if you are fuzzy on delta G's or equilibrium constants, go review this, because you are going to see a lot of this in this class, and Biochem 2 for that matter. So this is something that you should really try to refresh yourself on. Okay, so we're at the end, but what I want to really end with here is really an emphasis on the fact that what I have tried to do for this lecture is not go in great depth, but cover many of the big topics that we will see again in this class. I want to give this opportunity to you to sort of, put up a big sign that says, hey, know these topics, at least be familiar with them, so that when we use them to, advance biochemical concepts, it's not as though you're having to start 2 steps up. And what I mean by that is, if I need to explain something, a biochemical concept that is built from, say, thermodynamics or non-covalent interactions, I'm going to assume that you have some understanding of the foundational material, because otherwise we simply don't have time in this course to teach all of the Gen Chem topics as well as the biochemistry topics. So it is really going to be dependent upon you. You are going to have to invest the time to make sure that you are fresh on some of these concepts. Now, to give you the brief overview, here are some topics to review. This is generally an overall summary of what I have talked about here today. I told you about cells. I'm not a biologist, so I'm just going to leave it there. But if you want to go deeper, cellular architecture, compartmentalization, the context of biochemistry. I also talked about how awesome carbon is. Carbon can do a lot of things. It is versatile. It can form different kinds of covalent bonds. Functional groups. Functional groups are going to be critical to the formation of non-covalent interactions. So knowing what functional groups do, what they look like, the types of chemistries they can become involved in, that's going to be really important downstream. Having some familiarity with non-covalent interactions like hydrogen bonding, van der Waals interactions, hydrophobic effect, electrostatics, dipole-dipole interactions. These are going to be important because they are going to form the basis for biomolecular 3-dimensional structure. So non-covalent interactions are really going to be at the core of a lot of what we talk about here. And lastly, thermodynamics. I don't expect you to be an expert, but I want you to be able to interpret delta G, delta H, delta S, KQ. Know what they are, know what they mean. Have some sense for what happened, how to interpret a negative delta G versus a positive delta G. So, okay, that's it. The next lecture will begin in chapter 2, where we start talking about water. And so I'll see you then.